Hello guys, welcome back to another video. Today we'll be looking at cell biology, and today we'll be looking at part 1 of cell biology. Keywords. So we'll be looking at 6 keywords related to this topic. The first keyword is organelle, which is a structure within a cell. Number 2, mitosis. Mitosis is a type of special division that produces 2 identical daughter cells. 3, specialized cell. A cell that has features that allows it to carry out its function. 4, microscope. A microscope is used to enlarge very small objects that are not visible to the human eye, to the naked eye. Number 5, magnification is how many times larger an object is compared to its actual size. So it's usually times by 400, times by 100, so those are the common ones. 6, resolution. Resolution is how close together objects can be before they blur into one. So first we'll be looking at eukaryotes and prokaryotes. So cells of bacteria are called prokaryotic cells. On the other hand, cells of animals, fungi, and plants are called eukaryotic cells. So here's a table comparing prokaryotic eukaryotic cells. So the size of eukaryotic cells are mostly 5 to 100 micrometers. On the other hand, for prokaryotic cell, it's usually 0.2 to 2 micrometers. So that means that eukaryotic cells are much larger than prokaryotic cells, mainly because prokaryotic cells are mainly single-celled organisms. Outer layers of cells. Eukaryotic cells. They have a cell membrane surrounded by cell wall in plants and fungi. And in animal cells, they don't have a cell wall. On the other hand, prokaryotic cells have a cell membrane also surrounded by a cell wall. Cell contents. So eukaryotic cells, so they have cytoplasm and organelles. Cell organelles include mitochondria, chloroplasts in plants and ribosomes. Prokaryotic cells have a cytoplasm, ribosomes, but they don't have mitochondria or chloroplasts. On the other hand, genetic material. So in a eukaryotic cell, its DNA is in a nucleus and plasmids are found in a few small eukaryotic organisms. On the other hand, for prokaryotic cell, DNA is a single molecule found free in the cytoplasm. So type of cell division in a eukaryotic cell is called mitosis. On the other hand, for a prokaryotic cell, it's called binary fission. So animal cell. So the structures of an animal cell, so organelles they have a cytoplasm, which is a jelly-like material that contains dissolved nutrients and salts and structures called organelles. It is where many of the chemical reactions happen. So nucleus contains genetic material including DNA, which controls the cell activities. Cell membrane. Its structure is permeable to some substances but not to others. It therefore controls the movement of substances in and out of the cell. Mitochondria. So there are organelles that contain the enzymes for respiration and where most energy is released in respiration. Ribosomes are tiny structures where protein synthesis occurs, which is the creation of protein. For a plant cell, so they have 3 more structures than an animal cell. So they also have a nucleus, which contains genetic material and controls activities of the cell. They have a cytoplasm, it's where reactions happen. They have a cell membrane, which controls what moves in and out of a cell. They also have mitochondria, where aerobic respiration takes place, and ribosomes, which is a site of protein synthesis. In addition, they have a cell wall, which strengthens the cell, chloroplasts, which contain chlorophyll, and is a green pigment which allows them to photosynthesize. Finally, they have a vacuole. They have something called vacuole, which is filled with cell sap to keep the cell turgid and strong. So here are some animal cells and how they're adapted to their function. So sperm cell, the head of the sperm contains a genetic material for fertilization. The acrosome in the head contains enzymes so that the sperm can penetrate an egg. The middle piece is packed with mitochondria to release energy needed to swim and fertilize the egg. The cell enables the sperm to swim. So another one is a nerve cell. So the nerve cell is extended so that the nerves can run to and from different parts of the body to the central nervous system. So there are multiple nerve cells in our body. The cell has extensions and branches so that it can communicate with other nerve cells, muscles, and glands. It is covered with a fatty sheath which insulates the nerve cell and speeds up the nerve impulse. And finally, muscle cells. They contain filaments of protein that slide over each other to cause muscle contraction. The arrangement of these filaments causes a banded appearance of heart muscle and skeletal muscle. They contain many well-developed mitochondria to provide a lot of energy for muscular contraction and movement. So here are some examples of plant cells and how they're adapted to their function. So plants have root hair cells, which has a large surface area to provide contact with soil water, and it has thin walls so as not to restrict the movement of water. So if they have thin walls, that means the water can't really get out. And if it's big, so they have xylem cells. There are no top and bottom walls between xylem cells. So there is a continuous column of water running through them. The walls become thick and woody. They therefore support the plant. So they carry water and some dissolved minerals from the roots upwards to other parts of the plant. So this water evaporates and is lost from the leaves as water vapor during the continual process of transpiration. And transpiration is a gradual release of water vapor from leaves so that they can pull water up from the soil. And they carry a lot of water to the green plants. Green areas plant for photosynthesis during the day. In addition, they're made from lots of individual cells that have died and have no end walls. And finally, phloem cells. So dissolved sugars and amino acids can be transported both up and down the stem. Companion cells adjacent to the sieve tube provide energy to transport substances in the phloem. So mainly they carry glucose as sucrose made in photosynthesis from the leaves of the plant to all other parts of the plant in the cell cycle. So this process is translocation and is a movement of sugars made in photosynthesis from the leaves of plants. The sugar is used immediately in respiration to release energy for the plant, or is stored as starch in the cell, or in structures such as roots of vegetables. So unlike a xylem, these are living organisms. And they are specialized end walls called sieve plates, that have small holes to them. So usually phloem cells and xylem cells, they bunch up. They form bundles, and these are what form the veins in a leaf. But in a vein, there could be lots and lots of xylem and phloem cells. So now we'll be looking at light microscopes, mainly talking about combine microscopes. So if you think of a general animal cell, so they're between 0.01 to 0.05 millimeters. But plant cells are a little bit bigger, from 0.01 to 0.1 millimeters. The human eye can see objects as small as around 0.05 millimeters. A microscope is required to see cells in any detail. So microscopes magnify the image of a specimen so that it appears larger. So the magnification of a microscope is magnification of eyepiece times the magnification of objective. But the magnification of an image is the size of image divided by real size of image. So the resolution of a light microscope is around 0.01 2 micrometers or 200 nanometers. This means it cannot distinguish 2 points closer than 200 nanometers. On the other hand, we'll be talking about electron microscopes, which use a beam of electrons instead of light rays. There are two types of electron microscopes. So the scanning electron microscope, SEM, has a large field view, so can be used to examine the surface structure of specimens. So SEMs are often used at lower magnification. On the other hand, the transmission electron microscope is used to examine thin slices or sections. TEM transmission electron microscopes have a maximum magnification of around 1,000,000, but images can be enlarged beyond that photographically. And in a TEM, the limit of resolution is less than 1 nanometer, which means that it cannot distinguish 2 points closer than 1 nanometer. So if we compare the images from a transmission electron microscope and a scanning electron microscope, images from transmission electron microscopes are flat and usually taken cross-section to a specimen. That is, they are frequently used to look at a section to a cell. Scanning electron microscopes don't need thin samples, so they can use to make images that look more 3D. But all electron microscopes images are black and white. But the scientists sometimes color these images to make them more realistic. Thank you for watching my video. I hope you enjoyed it. Please like and subscribe to my channel, and look out for my other videos, including cell biology part 2 and part 3.